Bioremediation is the use of biological agents like bacteria and plants to remove or neutralize contaminants from polluted soil or water. There are two types of bioremediation. The first one is in situ, where we treat the contamination on the spot, and the second one is ex situ, where we treat the contamination away from the original spot. So today, we are going to discuss on a journal entitled with a mini review, Bioremediation Mechanism of Combined Pollution of PAH and Heavy Metal by Bacteria and Fungi. PAH stands for polycyclic aromatic hydrocarbon, chemical that often found together in a group of two or more and generally contain more aromatic ring and more complicated molecular structure. They are usually stable in environment. On the other hand, heavy metal is a metallic chemical element that has relatively high density and is toxic at low concentration. Different heavy metal will show various toxic effects.
According to this journal, the combined pollution of these two substances have widely spread to our environment and brought many harm effects to the earth, and living things include human beings and plants. Even they are needed in a small amount, they still can be hazardous in slightly larger than the amount required, as they have harmful chemicals such as carcinogenic, teratogenic, and mutagenic properties. This type of pollution is a contribution from industrial side and resulted from incomplete combustion of organic matter. Scientists found a way to treat this type of pollution by bioremediation and phytoremediation, as they are environmentally friendly and economical, as they are cost-effective technologies.
So, this journal will be discussing on the factors affecting biodegradation of PAH and heavy metal, the outline of bioremediation mechanism of PAH and heavy metal by bacteria and fungi, and lastly, the further exploration of PAH and heavy metal pollution. The bacteria and fungi are isolated from contaminated sediment and soil. Bacillus subtilis, E. coli, and Mycobacterium are commonly found in PAH and heavy metals. Moving to the first objective of study, the factors affecting biodegradation of PAH and heavy metals are environmental factor, microbial activity, and matrix effect.
Environmental factor can be divided into three factors, which are pH, temperature, and low molecular weight of organic acid and humic acid. pH is important to microbial organisms as different species require different pH level for living, and by adjusting the pH level at the polluted site, it could be a great attempt to increase the efficiencies of bioremediation of this pollution. While temperature is a crucial factor in affecting biodegradation of PAH and heavy metal, as the solubility of heavy metal increase when the temperature is increased. In a way, it will increase the bioavailability and accelerate bioremediation process as it influences the absorption and absorption of contaminant on microbes.
The second factor is microbial activity. The species of microbes used must be screened to check on the genes that might affect the microbial activity. The strain screening from contaminated area usually show high capacity to tackle with pollution of PAH and heavy metal. The DNA located in strain with high capacity of repairing PAH and heavy metal contain resistance genes or destruction genes, which can be separated using genomic approach to enhance the efficiencies.
The last factor that contributes to this degradation is matrix effects, concentrated on the impact of concentration, structure, solubility, and absorption of PAH and heavy metals. There are three types of interaction, which are heavy metal with heavy metal, PAH with PAH, and PAH with heavy metal. The first one is heavy metal with heavy metal, which some heavy metal are fundamental for biological processes of the microbes, whereas others bring no biological benefit but only inducing oxidative damages, denaturing organisms, and weakening the bioremediation ability of microbes to heavy metal, which are the first one is alter physiological and biochemical properties of microbe. Second, pose deleterious effect to microorganism. Third, break the vital enzymatic function. Fourth is cause ion imbalance, and the last one is directly cause DNA damage or affect enzyme formation. In return, heavy metal stimulate the antioxidant defensive system of microbes.
The second interaction is between PAH and PAH. PAH metabolites can impact the degradation of PAH. It is found out that enterics, fluorine, and pyrines could not be degraded by Micrococcus sp, but with the addition of naphthalene and phenanthrene, all of them could be degraded with the degradation rates of more than 90% over 10 months incubation. The intermediates of PAH could also have an effect of the degradation of PAH, acid intermediation may be unsuitable or the concentration are too high for the growth of microbes, thus suppressing the pH degradation.
The third interaction is between heavy metal and PAH. Heavy metal can affect PAH degradation by changing the surface properties of microbe and interfering the enzyme of microbe. Heavy metal also can affect the absorption of PAH by microbe via changing the surface of microbe. Heavy metal stress will cause oxidation process by ROS, which is a reactive oxygen species, which will impair membrane and stimulate the development of antioxidant enzymatic. So, do we agree with this method for treating PAH and heavy metal?
Yes, we do agree. Bioremediation method use living organisms such as bacteria and fungi to treat pollution and contaminants. Therefore, this method is perceived by the public. It is also an environmental friendly method because it can be done without causing major disruptive to the environment. It is also cost-effective technologies.
According to other journals, this method is the most preferred method used to treat contaminant. This method is easy to conduct by using either in situ or ex situ. Even if there are certain parties claim that this method is not very effective to treat pollution because of the few side effects, it will still not cause any big damages to our environment.
However, further research must be done to study more on how efficient and effective of this method to treat pollution in our environment so that we can give awareness to the public about this treatment, including the specific species of microbe that can be used for this treatment and the side effect of metabolic substance released by the microbes. Also, suitable method to enlarge the viability that may increase the remediation effect. I'm Afika binti Azhar.
And I'm Latifah binti Rashid.
I'm Arfa Fadin Hazika binti Jamali.
We are from AS 1145A1 and thanks for watching.